Dear everyone, today we are talking about post-transcriptional modification. Post-transcriptional modification is also known as RNA processing. What is RNA? RNA is a polymer of nucleotides which are synthesized from DNA, and the synthesis of RNA molecule from DNA is known as transcription. After being transcribed a nascent RNA molecule of eukaryotic organism, this nascent RNA molecule must undergo post-transcriptional modification because this hnRNA, that is heteronuclear RNA molecule, contain several non-coding sequences which is known as intron, which is not required, meaning thereby introns, that is the non-coding sequences, are not required for protein synthesis. That's why this non-coding sequence introns must be removed from hnRNA. So, post-transcriptional modification is required for stability and integrity of newly synthesized RNA molecule, that is mRNA. And newly synthesized RNA molecule is known as hnRNA, heterogeneous nuclear RNA. Heterogeneous nuclear RNA contain both coding and non-coding sequence. Coding sequence is known as exons and non-coding sequences are known as intron. So, introns must be removed from hnRNA. This is one type of post-transcriptional modification. Second thing occurs, the capping process. 5 prime must be modified. There is the chemical modification at 5 prime. That chemical modification is known as 5 prime capping. And at the 3 end, that is the 3 prime, there is addition of several adenine molecule, that is known as polyadenylation. So, this RNA processing or post-transcriptional modification are of 3 types. One is splicing. Splicing means removal of non-coding sequence from hnRNA. Second one is 5 prime capping, and third one is 3 prime polyadenylation. So, in this video, we have to discuss these 3 type of modification which occur after transcription and before translation, and this post transcriptional modification occurs only in eukaryotes. This not occur in prokaryotic cell. So, post transcriptional modification in eukaryote involve 3 major type of modification. First one is splicing. Splicing means a removal of introns from nascent RNA, that is hnRNA. Second one is capping at 5 prime. Methyl residue is added at 7 nitrogen in guanine, that is the M7G cap. That's why here is written 7 M7G cap. This is the capping at 5 prime. And third one is polyadenylation. There is the incorporation of poly A tail at 3 prime, 3 prime. And one more modification is very specific, and this modification is known as RNA editing. RNA editing will be discussed in a separate video. Here we have to understand splicing, capping and polyadenylation. So look at this slide. You can see a newly transcribed hnRNA contain intron, exon. Alternatively, there is the arrangement of intron and exon. As we know, intron is the non-coding sequence which is not required in protein synthesis. That's why in nucleus this new intron is removed out from the hnRNA, and this removal of intron from hnRNA is known as splicing. Thereafter, there is certain modification, that is the chemical modification at 5 prime, which is known as capping at 5 prime, and second modification is polyadenylation, in which there is the addition of about 200 to 300 adenine residues at 3 prime of newly synthesized hnRNA. This addition of poly A is known as polyadenylation, and at 5 prime there is the 5 cap, which protects the nascent mRNA from degradation and helps in ribosome binding during protein synthesis during the translation process. So this all three modifications which occur after transcription is completed. So this was all about the post-transcriptional modification. Post-transcriptional modification occurs only in eukaryotic organism but not in prokaryotic cells. Thanks for watching.